Hi students, today I am going to discuss about nutritional biochemistry. Before going that, you have to know classification of amino acids and protein, and that start from the introduction. Living cell produce an impressive variety of large molecule, particularly proteins, nucleic acids and carbohydrate. They serve as structural component, biocatalyst, hormones, receptor or repositories of genetic information. These large molecules are biopolymers constructed of monomer units. For proteins, the monomer units are amino acids. Therefore, to comprehensive protein chemistry, some knowledge of chemistry of amino acid is essential. Now, we can go start with objective. To recall the meaning of amino acids, to list down the method of classification of amino acids, to define protein, to categorize the types of protein, to describe the structure of amino acids and protein. Now, we can discuss about amino acids, and then we start from the definition. Amino acids are a group of organic compounds containing two functional groups. One is amino acids, another one is carboxyl. The amino group is NH2 is a basic, while the carboxyl group is COOH. Both are in acidic in nature. We can discuss about classification of amino acids. There are different ways of classifying the amino acid based on the structure, chemical nature, nutritional requirement and metabolic fate. First, we will go discuss with one by one. The first one, amino acid classification based on the structure. The comprehensive classification of amino acid is based on their structure and chemical nature. Each amino acid is assigned a 3-letter or 1-letter symbol. These symbols are commonly used to represent the amino acid in protein structure. The 20 amino acids found in proteins are divided into 7 distinct groups. The first one, amino acids with aliphatic side chain. These are monoamino monocarboxylic acids. This group consists of most simple amino acids like glycine, alanine, valine, leucine and isoleucine. The last 3 amino acids, leucine, isoleucine, valine, contain branched aliphatic side chain, hence they are referred to as branched chain amino acids. The second one, hydroxyl group containing amino acids. Serine, threonine and tyrosine are hydroxyl group containing amino acids. Tyrosine being aromatic in nature, so it is undercome aromatic amino acids. The third one, sulfur containing amino acid. Cystine and methionine with thioether group are the 2 amino acids. Cystine is another important sulfur containing amino acid, is referred by the condensation of 2 molecules of cystine. Aromatic amino acids and their amides. Aspartic acid and glutamic acids are dicarboxylic monoamino acids, while asparagine and glutamine are the derivative of amides. The next classification under structure, basic amino acids. The 3 amino acids, lysine, arginine with guanidino group and histidine with imidazole ring are dibasic monocarboxylic acids. They are highly basic character. The next one, aromatic amino acids, phenylalanine, tyrosine and tryptophan. Tryptophan is containing indole ring or aromatic amino acids. Besides, histidine may also consider under this category. The next one, amino acids, proline containing pyrrolidine ring is a unique amino acids. It has an amino group double bond NH instead of the normal amino group NH2. Therefore, proline is an amino acids. The next classification is based on the polarity. Amino acids are classified into 4 groups based on their polarity. The polarity in turn reflects the functional role of amino acids in structure. From that, the first one, non-polar amino acids. These amino acids are also referred to as hydrophobic, water hating. They have no charge on the R group. The amino acids included in this group are alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan and proline. Polar amino acids with no charge on R groups. These amino acids as such carry no charge on R group. However, it possesses groups such as hydroxyl, sulfhydryl and amide and precipitate in hydrogen bonding of protein structure. The simple amino acid glycine, where R equal to H, is also referred in this category. The amino acids in this group are glycine, serine, threonine, cysteine, glutamine, asparagine and tyrosine. Another one, polar amino acids with positive R group. The 3 amino acids lysine, arginine and histidine are included in this group. Next one, polar amino acids with negative R group. The dicarboxylic monoamino acids, particularly aspartic acid and glutamic acids, are considered in this group. From the third classification, nutritional classification of amino acids. The 20 amino acids are required for the synthesis of variety of proteins besides other biological functions. However, all these 20 amino acids need not to be taken in the diet. Based on the nutritional requirement, amino acids are grouped into 2 classes. One is essential amino acid and non-essential amino acid. From the first one, essential or indispensable amino acids. The amino acids which cannot be synthesized by the body and therefore need to be supplied through the diet are called essential amino acids. They are required for proper growth and maintenance of the individual. The 10 amino acids are arginine, histidine, threonine, phenylalanine, tryptophan, leucine, lysine, isoleucine, valine and methionine. Non-essential or dispensable amino acids. The body can synthesize about 10 amino acids to meet the biological needs. Hence, they need not be consumed in the diet. These are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine and proline. The fourth one, amino acids classification based on their metabolic fate. The carbon skeleton of amino acids can serve as the precursor for the synthesis of glucose. From metabolic viewpoint, amino acids are divided into 3 main categories. One is glycogenic amino acids. These amino acids can serve as precursor for the formation of glucose or glycogen. Example, alanine, aspartate, glycine, methionine. The next one, ketogenic amino acids. Fat can be synthesized from these amino acids. Only 2 amino acids are exclusive. These are leucine and lysine. The third one, glycogenic and ketogenic amino acids. The 4 amino acids isoleucine, phenylalanine, tryptophan and tyrosine are the precursor for the synthesis of both glucose as well as fat. Now we can go to the next module, is the peptide bond. A peptide bond is chemical bond formed between 2 molecules. Formation of how the peptide bond is formed. When the carboxyl group of one molecule react with amino group of other molecule, they are releasing a water molecule, that is H2O. This is the dehydration synthesis of reaction. It is also known as condensation reaction and usually occur between 2 amino acids. After the peptide bond, you have to know what are the characteristics of the peptide. The peptide bond is rigid and planar with partial double bond in character. It generally exists in trans configuration. Both the C double bond O and NH groups of peptide bond are polar and are involved in hydrogen bond formation. And then we have to know the biological importance of the peptide. Several peptides occur in living organisms that display a wide spectrum of biological function. Generally, the term peptide is applied when the number of constituent of amino acid is less than 10. Some examples: biologically active peptide, and these functions you have to discuss. Biological aspects of peptide. The one example is glutathione. It is a tripeptide composed of 3 amino acids. Chemically, it is glutamyl, cysteine and glycine. It is widely distributed in nature and exists in both reduced and oxidized state. The next one is thyrotropin releasing hormone. It is also a tripeptide secreted by hypothalamus. It stimulates pituitary gland and release thyrotropic hormone. And the third one, oxytocin. It is an hormone secreted by posterior pituitary gland and contains 9 amino acids. It is otherwise called nonapeptide. Oxytocin causes contraction of uterus. Another hormone, vasopressin, is otherwise called antidiuretic hormone, ADH. ADH is also nanopeptide produced by posterior pituitary gland. It stimulates kidney to retain water and that increase the blood pressure. And another one, angiotensin. Angiotensin 1 is a decapeptide, that means 10 amino acids, which is converted to angiotensin 2 from 10 to 8 amino acids. The latter has more hypertensive effect. Angiotensin 2 also stimulate the release of aldosterone from adrenal gland. And then you can go discuss with the protein. From that module, from the nutritional biochemistry, you have to know the classification of amino acids and protein. First you finish the classification of amino acids and their function. Now we can start with protein. Before that, you have to know the definition. Proteins are polymer of amino acids produced by living cells in all form of life. A large number of proteins exist with diverse function, particularly size, shape and structure, but each is composed of essential and non-essential amino acid in varying the number and sequence. The number of distinct protein within one cell is estimated at 3000 to 5000. A typical protein contains 200 to 300 amino acids, but some are much smaller and some much larger. For example, you can take an insulin. The molecular weight of the protein is 6000 kilodalton. It goes to millions. That is under structural protein. Classification of proteins. The classification of proteins is divided into 3 main categories. One is simple protein, conjugated protein and derived protein. The simple protein is further divided into 2 categories, globular protein and fibrous protein. From that one, we can start from globular proteins. The globular proteins are spherical or oval in shape. It is soluble in water or other solvent and easily digestible. First one, albumin. It is soluble in water. It is coagulated by heat and precipitated by high salt concentration. Example, serum albumin, lactalbumin and leucocin. And another one, globulins. It is insoluble in water, soluble in dilute salt solution and precipitated by half saturated salt solution. Example, serum albumin, vitelin, tuberin, myosinogen. Next one, glutalin. It is insoluble in water, but soluble in dilute acids and alkali. It is mostly found in plants. Example, glutenine and oryzanine. Prolamines, it is insoluble in water and absolute alcohol, but soluble in 70 to 80% of alcohol. Example, gliadin and zein. Protamines, it is a basic protein, low molecular weight. It is soluble in water, dilute acids and alkali. Example, salmine. From the simple protein, the next one is fibrous protein. These are fiber-like shape, insoluble in water and resistant to digestion. The example, collagen. These are all connective tissue protein. It is a lacking of tryptophan. Collagens on boiling with water or dilute acids, it yield gelatin which is soluble and digestible. The next one, elastin. It is found in elastic tissues such as tendon and arteries. Keratin, it is present in exoskeletal structure. Example, hair, nails and horns. Human hair contains as much as 14% cysteine. The next one of classification of protein, conjugated protein. The conjugated protein is based on the prosthetic group is the main role. The first one is nucleoprotein. The nucleic acid, particularly DNA, RNA is the prosthetic group. Example, nucleohistone and nucleoprotamine. Glycoprotein, the prosthetic group is carbohydrate which is less than 4% of protein. The third one, lipoprotein, a lipid is the prosthetic group. Example, serum lipoproteins and membrane lipoproteins. Chromoprotein, the prosthetic group is colored in nature. Example, hemoglobins and cytochrome. Metalloprotein, these protein contains metal ion such as iron, cobalt, zinc, copper and magnesium. From the classification of protein, the third one is derived protein. The derived proteins are again is divided into two categories. One is primary derived protein which are denatured or coagulated. The secondary derived proteins are degraded due to breakdown of peptide and the product of proteins. You know about the classification of protein, then how it is organization. That means you know the structure of protein. From that organization of protein is divided into four categories, namely primary, secondary, tertiary and quaternary structure. The primary structure is the sequence of amino acids, that is called primary structure. It is a linear chain. The secondary structure is the conformation of polypeptide chain by twisting or folding is referred to as secondary structure. The amino acids are located close to each other in their sequence. The secondary structure are divided into two types. One is alpha helix and beta sheet. The alpha helical structure was proposed by Pauling and Corney in 1951. It is tightly packed coil structure with amino acid side chain extending outwards from the central axis. The alpha helix is stabilized by extensive hydrogen bonding. It is formed between hydrogen atom attached to peptide nitrogen and oxygen atom attached to carbon. All the peptide bonds except the first and last in a peptide chain participate in hydrogen bonding. Each turn of alpha helix contain 3.6 amino acids and the distance between these is 0.54 nanometer. The space between each amino acid is 0.15 nanometer. The next one, beta sheets, which is also proposed by Pauling and Corney. Beta sheets are composed of two or more segments or fully extended peptide chain. The hydrogen bonds are formed between neighboring segments of polypeptide chain. The polypeptide chain may be arranged either in parallel or anti-parallel. Beta pleated sheets may be formed either by separate polypeptide chain or the single polypeptide chain folding back on itself. That means the between hydrogen bond interaction. The third one, tertiary structure. The three-dimensional arrangement of protein structure is referred to as tertiary structure. It is a compact structure with hydrophobic side chain held interior, while the hydrophilic groups are on the surface of the protein molecule. Besides, the hydrogen bonds, disulfide bonds, that means sulfur and ionic interaction and hydrophobic interaction also contribute to the tertiary structure. The last one, quaternary structure. The protein consists of two or more peptide chain held together by non-covalent interaction or by covalent cross linkage. Now you understand that the organization of protein, primary, secondary, tertiary and quaternary structure. From this nutritional biochemistry, particularly this module, you understand that the classification of amino acids and protein. The amino acids in the form of protein perform structural, hormonal and catalytic function. It is based on structure, chemical nature, nutritional requirement and metabolic fate. The essential amino acids are not synthesized in our body, it is available only in the diet. It is support to infant growth and maintenance of human health. Proteins are classified into three main groups. Simple protein contain only amino acid residues. Conjugated proteins contain a non-protein moiety, namely prosthetic group. Derived proteins are obtained from degradation of simple or conjugated protein. Now you understand the classification of amino acids and protein. In the next module, we will continue with the protein synthesis. Bye.